Mr. Dushyant Singh Baghel. Mr. Baghel is the co-founder and CEO of Nucleon Informatics, a leading genomics and bioinformatics company based at Hyderabad. His company has successfully sequenced and assembled genomes of mithun, mango, Lucinodes or bonalis, tiger, pomegranate, and many microbial and fungal genomes.
I have been sequencing genomes since 2007 onwards, and I would like to speak about few of the challenges that we have faced in recent fish genomes that we are doing with CIBA. Along with that, we will also capture a little bit information about Nucleon, then what all we have done in last 6 or 7 years, and then what should be the possible strategy for sequencing complex fish genomes, and how can you finish the genome assemblies using some 4th generation sequencing technologies and further data analysis and the validation of the assemblies. Nucleon was started in 2012-13. We have been, before that, we have been doing genome sequencing, working as a genome sequencing service provider for a company based in Ahmedabad. This is my second company. This is mostly involved in sequencing and assembly of the genomes, transcriptomes, metagenomes, and epigenomes. We also cover entire spectrum of metagenomics and epigenomics. We also have a very good data analysis support, and we have developed, with the help of Dell, a product called Big Data Decoder, which is a server mainly used for NGS data analysis. We have developed our own pipeline, which is under patent, called Dr. Seek. This is used for transcriptome data analysis and metagenome data analysis. We are very soon coming up with Dr. Seek de novo, which will be used for hybrid genome assembly. I have been involved in various genome sequencing programs for India, including Indian wild dog, goat genome, then one very prestigious project of buffalo genome sequencing as well, which is funded by the World Bank. Along with that, multiple plant genomes and smaller genomes like bacteria and viruses. These are the few clients that we have across the world. When we talk about genome complexity is extremely wide area. After doing at least 38 genome sequencing and assembly, I came to CIBA, and whatever knowledge I had gained in last 12 years of sequencing, everything was gone, because the moment I saw shrimp genome data set, extremely difficult. When you talk about, you want to start a genome sequence, you should know a few things before starting the genome sequencing and assembly. First is genome size, repeats, number of genes, heterozygosity level, contamination level, or structural variance possibility. So I'll just give you one basic example how this whole complexity can arise. We as human has 3.5 GB as almost 3 GB, 3 to 3.5 GB as haploid genome size, whereas a small parasite, which is T. vaginalis, has a genome size of 160 MB only. Genome of human carries around 20 to 25,000 genes, and 160 MB, just 160 MB of parasite carries 60,000 genes. We have one fish which is 130 GB long. So you can see how a small a genome like bacteria can be around 1 MB or 2 MB, virus can be in KBs. So how complex the whole genetic system is. So before doing any genome sequencing, we should know what is the genome size, what sort of number of repeats we have, so do some genome survey before jumping on the genome sequencing and assembly programs. So what we recommend to our clients is always start with a strategy first. Don't jump on writing the projects just because you want to write. Start working with bioinformatician, find out what all is happening across the world so that you can build a strategy. Start with the sample prep, libraries, sequencing, what sort of sequencing do we need, how much coverage do we need, how many technologies we have to use. In case of data analysis, what sort of hybrid assembly approaches we can use. So we build the project execution strategies, and the best part of sequencing is DNA. Most of you can isolate genomic DNA, but we are looking for a very high molecular weight genomic DNA, which is around 300 kb. If you can get 300 kb genomic DNA from your sample, it's very easy to utilize 4th generation genomic solution, which can use for finishing the genomes. So in case of shrimp, we faced a huge problem because of the repeats, and even the isolation of genomic DNA was not easy. So we standardized few of the kits and found that QIAGEN MagAttract high molecular weight genomic DNA kit, along with some cleaning steps, works for the shrimp. If you want to do the genome survey, I would recommend you should use for Illumina TruSeq PCR free library, generate some data set on Illumina TruSeq PCR free library, and do some genome survey. Find out how much data, do some k-mer analysis, repetitive analysis, heterozygosity level, find out these information. These information would be very helpful in choosing the pipeline and further deciding which chemistry you have to use for genome sequencing. Usually we recommend our clients to use PacBio. You can, other than if you don't have much funds, you can also opt for Nanopore. Nanopore is also coming up very well. In case of PacBio, I would always recommend to go with larger read length and the largest read length possible. So for larger read length, you need a very large insert size library. So opting for 30 to 40 KB library should be sufficient for a good shrimp genome or fish genome sequencing. In case of library preparation, there is a hitch. It's always good to start with a very high molecular weight genomic DNA, but the size selection is extremely important. We are looking at some of the fragments which should be around 25 to 75 KB. You should not select less than 25 KB of fragments. The loading also plays very important role. If you load much, you may come up with some of the reads which will be giving you the sequences from same region. If you want to cover the entire sequences, if you want to cover the entire genomic content of the species, you should go with very large insert size, and the possibility of size selection cannot be neglected. Sequencing on SQL system. If you talk about only PacBio, having 60x coverage on PacBio is sufficient for any deployed genome. But if you are looking for a very highly repetitive genome, 80 to 100x coverage should be sufficient. I'm not sure about the fishes will have polyploidy or not. So if it's a polyploid genome like some of the plant samples, then you should opt for around 120x coverage. Genome assembly on PacBio dataset is extremely easy. You can opt for either a smart link, which is developed by PacBio itself, or you can also opt for command line tools if you have your own bioinformatics team available. If you don't have the bioinformatics team, don't worry. We have a very good bioinformatics team in Hyderabad itself. You can always contact us and we can help you with the data analysis. After assembly, it's very important to polish the genome. Polishing is the step when you want to take away the sequences which are not of good quality, the bases which are of not good quality. You want to ultimately improve the quality of the assembly by polishing step. You can use Skewer and Arrow algorithms which are freely available. So what is beyond draft genome assembly? That is a major question. So why I opted for complex fish genomes? Because fish genomes, when I started doing shrimp genome, I was not aware of how complex the fish genome can be. So even the normal genomic DNA isolation step doesn't work. Library preparation, the recovery ratio doesn't work. Usually recovery in case of normal plant and animal sample, if you take 5 microgram of genomic DNA, you will come up with at least 800 nanogram or 1 microgram of library. But in case of shrimp, it is too low. It is around 100 to 200 or even sometime lower. So you have to be very careful while isolation as well as library preparation. Just because you are dealing with single individual, you cannot change, you cannot have more number of individuals, otherwise that may increase the heterozygosity. You have to opt for a very accurate technology which can give you high molecular weight genomic DNA, and as well as you should have you have to standardize the library loading as well. But once you generate the data, assemble the deployed genome, you get the contigs. These contigs are very small fragments, consensus which has been built by a software, but these are not your chromosomes. You should have some other methodology to build chromosomes. So this slide I have taken from Adam Philippi. This slide was presented in Vertebrate Genome Program. They have also observed a similar strategy. They are also sequencing around 10,000 different vertebrates. So they are also starting with PacBio to build the context. They are followed by the 10x genomics for scaffolding, phasing, and polishing of the genome. After that, super scaffolding with Bionano and validation of the assembly as well. For building the chromosome level scaffolds and phasing out the genomes, we are also opting for Hi-C. The similar thing we have done. So what are the scaffolding options you have as of now? If you are building a very high quality quantic assembly which is larger than 1 MB, this is a suitable, you can opt for made pair data for Illumina and can build the scaffold. If you don't have 1 MB quantic N50 and you are looking for some other approaches, you can either go for 10x genomics, build supernova assembly, and use SCAF 10x and build hybrid assembly. You can also opt for Bionano, which can give you up to 300 KB links and can generate the Bionano based genome map, which is a good way to assemble the genome. This Bionano can be used for the standalone genome assembly also. If you don't want to use any other tool, you can use standalone Bionano for building the genomes. If you want to use Bionano for scaffolding, you can use genome map along with the 10x genomics and the Chicago and Hi-C data sets as well. Chicago is from Dovetail. You can use this technology for Hi-C data analysis or Hi-C based super scaffolding and chromosome confirmation. So I'll just quickly go through some of the technology which are available for complex genomics nowadays. If you are looking to sequence a fish genome, which is a fish which is very, very small, a tiny fish. So starting material is the problem, right? So in this case, you can opt for 10x genomics because the starting material that we need is only 1 nanogram. So this can help those who are looking for very small fishes. This can be used for generation of the data for the scaffolding as well. I'll quickly jump on what all has been achieved using 10x genomics data and supernova assembly. You can see in case of human, we have a very high quality assembly with N50 of 16 MB, and the entire genome has been assembled in very less number of scaffolds. In non-human cases also, chili pepper, monk seal, hummingbird, spotted owl, dragon, all these genomes have been assembled using only 10x genomics, sequencing on Hi-C and supernova assembly. Similar approaches can be used for sequencing sequencing genomes which are very large as well. Bionano optical mapping is not sequencing. It's totally different way of analyzing the genomes. You have to isolate a very high molecular weight genomic DNA which is larger than 300 KB. Once it is available, and you have a closely related genome or the genome which you have recently built, you can use that information and create a NICI report, which is nothing but identifying the enzymes which can nick the, it's nothing but the endonuclease identification which can nick in 100 KB, how many nicks it can create. So you identify the endonuclease which can create at least 6 to 7 nicks in 100 KB, use that for labeling and nicking, and then further labeling and optical mapping on sapphire chips. Bionano has its own software called Bionano Access. You can use this software for building the assembly and further identifying the structural variants against the reference genome or closely related genomes or against the genome map versus the NGS assembly. By Bionano optical map, you can generate de novo consensus sequences and which are very long. Usually you can get almost every time whenever I have used optical map on Sapphire, my N50 was always beyond 10 MB. So if you are struggling with low N50 values, you can always opt for optical mapping. These approaches have been used all across the world and more than 270 unique genome sequencing genome have been analyzed using Bionano, Iris and Sapphire system. Recently, I was invited in San Diego to present my work on PromoCananet, and there also we got a very excellent genome assembly, which is around 16 MB N50, and we assembled the entire genome of 364 MB, and which is covering almost 98% of the genome with BUSCO a score of 97%. There we realize how important it is to use optical mapping. So you should not only rely on sequencing methodology to sequence the genomes. You can use some other methodology like mapping approaches and Hi-C methods for genome sequencing and confirmation. Hi-C plays extremely important role if you are looking for finishing of the assembly. There are few prerequisites before jumping on the Hi-C. First is you should try other methodology to improve the assembly quality. Try to find out, let's say if you are working on a shrimp genome And recently, one of the shrimp genome is published in Nature Communication on 21st of January. This genome has 2.45 GB of size, and assembled size is only 1.6 GB with N50 value, counting N50 value of 50 KB. If you look at the entire gap, we have almost a gap of a GB, which is yet to be assembled. It's only because that we have all the genome is full of repeats. So you do not have the technology to resolve the repeats. So the only technology that we have as of now is PacBio based technology or Bionano or 10x genomics. So use hybrid assembly approaches to build assembly which is very close to the genome. Try to decrease the size of the scaffolds and increase the scaffold N50. These are, once these strategies are at, you are at the level of, at the satisfactory level where you are okay with it, then you can jump for high C. High C is nothing but you isolate the chromatids, you cross-link them, fragment them, and then ligate them and use them for sequencing on high-seq system. Generally, we recommend around 60 to 70x coverage on high-C. If you have a very complex genome, you can opt for at least 100x coverage. Other than this, I'm not sure about fishes, whether with all the fishes, I mean, experts can guide us. But recently, we have been working on a plant genome called mango with ICR. And there we are using trio-binning approach in case of plant. This trio-binning approach is nothing but, in case of mango, we are sequencing Amrapali genome with Dr. N.K. Singh ji. And the Amrapali's parents are, I think so, Dashari and Neelam. These two parents are sequenced at 50x coverage, and then followed by this, we also have the PacBio reads of Amrapali, which is nothing but the offspring. In this case, this slide is from my friend Sergi Koren, who has developed the trio-binning approach in case of these cattle genome, as well as we are doing the same approach for buffalo. So in case of mango genome, we are trying to separate these two parental haplotypes and by matching the k-mers. And then finally assembling these matched k-mers for individual haplotypes assembly. Finally, the ultimate assembly can be presented as diploid assembly. This is also a very good approach. If you can generate mama, papa, and offspring in case of fishes, you can use trio-binning approach. It's very economical and easiest method for assembly of highly heterozygous and complex genomes. So as you know, short reads are not like large reads, and they may miss some large variations. So it's very important that you can cover your entire data with a mix of short reads and large reads as well. Long read polishing is extremely important step in case of hybrid assembly programs, because you cannot always come up with assembly which is always correct. There will be some errors. You have to resolve the errors by polishing step. Haploid assembly, to summarize the entire thing, I would say haploid assembly is solved by long reads. You can easily build a haploid assembly by long reads. Then as soon as the reads are getting cheaper and economical, you can use different technology for sequencing. Nanopore can be also one possible solution, because PacBio is a little bit costly. Then it's very difficult to build diploid, polyploid, and complex genomes. So you should have faith on your bioinformatician. You should give some time to them so that they can build the entire information for you. There are some new tools which are getting developed. Please start using them so that you can build a nice and more informed genome assembly. I would like to thank Dr. Vijayan for giving me the opportunity to sequence shrimp genome, Dr. Shekhar for all the patience and multiple rounds of samples that you have given to us, sir. Dr. Vinay for all the misassemblies that we were trying to assemble. Then, and thank you so much for all your time during the analysis. I would like to thank my friend Sergi Koren for all the slides that he has shared with me regarding trio-binning, and Adam Philippi for sharing the slides for related to vertebrate genome program, Sarah Kingen for the PacBio sequencing related slides, my colleague Salu Yalapoina, who was involved in sequencing assembly, Satish Kumar, who was taking care of wet lab, Harish, the one who has assembled shrimp genome for us, Abhishek and Bipin Kumar, who are handling the projects. Thank you so much.